found this on my camera roll. I didn't even look at what it is. I figured it was fitting though.
Yeah, we all have something we are proud of, like that Western blot I have behind me.
Gorgeous.
I'll hold that with me forever.
Yeah.
Hey Maria, I thought you are from Poland, right?
Yes, yes.
Okay, Maria is not in our GroupMe because the GroupMe is not available in Poland, and I didn't know that.
My phone number didn't work. I don't know. They couldn't send me a verification code or something. So I'm sorry about that.
That's fine. That's fine. I'm glad you're joining us. Can you tell us something about yourself?
Well, I just graduated from my master's studies. As for now, I'm having a little bit of summer holidays. And as of October, I'm starting my PhD at my current university. So I'm kind of continuing in the same team, but I will be doing something totally different.
Okay.
Because the project finished and we are starting the next project.
That's pretty cool. By the way, let me tell you guys, we are live on YouTube. Okay, so just in case. And Harris, can you also tell us something about yourself again?
Yeah, absolutely. So I'm Harrison, third-year graduate student at the University of Georgia. I did some work previously in the biodefense space, and currently my research is on vaccine and therapeutic development for respiratory viruses, which is especially poignant nowadays with COVID. Our laboratory does a lot of COVID diagnostic and therapeutic development, but my thesis is revolved around respiratory syncytial virus vaccine development.
Oh, that's pretty cool stuff. Tanu is also Shutonuka. You can call her Tanu if that's easier.
That's easier.
Tanu, can you tell us about yourself quickly?
Yep, I am Shutanuka and I am a graduate student at the University of Florida. I'll be just starting 4th year. And I work with noroviruses and the microbiome and how their interaction affects the host immunological response and the host infection.
That's pretty nice. We got 2 virologists. The only person missing is the presenter for today, Anthea. She should be here. Do you guys have any experience with autoimmunity, SLE?
No.
No? Okay, that'll be a fun discussion.
No, but that's actually the field that I'm interested in. So
nice. Yeah, feel free to ask anything, even if you think it's a basic question, it's all right. Okay, just like I ask very simple things when it comes to infectious diseases, and even though it makes me look like a fool sometimes, it's fine. Anthea is an expert. Anthea is.
Let her introduce herself.
Yeah, Anthea, why don't you introduce yourself?
I'm sorry, I was like a bit late, you guys. I have
no problem at all.
Babies that I'm trying to mute. So if you hear little people running around, you know why. Okay. Is this lighting good for you guys? Can you see that?
Mm-hmm.
Yeah.
Yep.
Okay. How do I look?
Gorgeous.
Awesome.
10 out of 10.
Okay. So I am going to present today. I will admit on what is it? Figure 5. I think I'm gonna need some support from you.
Okay. I've got some support here with my notebook, but yeah, that's all the support I have.
Oh yeah. So I have my PowerPoint. How am I to do this, Jiten? Am I just going to upload?
I'll let you share your screen, and then I'll just make you a co-host. And Jeff.
Okay. How does my microphone sound?
It's audible.
You're good.
Okay. We're not live yet, are we?
We are live.
Yeah, live.
Yeah.
On YouTube?
Yeah.
Yes.
Yeah, we're live. You're 2 minutes late.
No, you're at the right time.
Put me on blast.
Good.
Okay. I'm going to make this bigger. Okay, so I'm sharing my screen. Oh, it says 1 participant at a time.
Am I sharing my screen? No. I'm not sharing my screen. Can you
Oh, okay.
check
I see.
again? Okay.
Okay. I think you guys see that.
Yeah, perfect.
Yes.
Like that. Okie dokie. Okay. Can you see that?
Yep.
All right. Let me know when you're ready.
We're ready. Actually, at least I'm ready. Everybody
Ready?
else?
Yep.
Yeah, we can start.
Let's go.
Okay. Let me get my notes. That's the most important part. Okay. So, good morning. I think for me it's morning. Hopefully, it's not like nighttime for anyone else, but good morning. I will be presenting today a paper titled Bcl6 suppresses miR, we'll call it miR, 142 3P-5P. I'll be using just miR-142 in future references. But this to express in SLE CD4 positive T cells by modulating histone methylation and acetylation of miR-142 promoter region. And this is by Ding et al. This is a group actually in China. I found this paper, one, just to tell a little bit about how this talk came to be today. But I found this paper because I'm actually reading the book. I don't know if you guys are familiar with, I think it's called Epigenetic Evolution.
Yeah.
Evolution of Epigenetics.
Mm-hmm.
Yeah, so oh my gosh, if you guys haven't read that book, and I'm still like not even halfway there yet, but it gets so exciting because it talks about just how they came to the point of understanding epigenetics, you know. So they did very, like, you would think simple experiments, but they're actually way more impactful than we like gave credit to today. They've looked at taking like embryonic nuclei and putting it into adult cells and seeing if it actually changes the functions of those cells. So they saw that the environment plays a heavy influence on what the genes actually say, right? Because we know all cells in our body has genetic information, you know, but then you think about it, it's like, how are these cells only specialized to be skin cells, or how are these cells only specialized to be T cells? And what this book is referring to is that all cells have the same genetic information, but as they mature or they become differentiated in different environments, they actually suppress certain, or permanently suppress gene activity so that they only act like T cells, or they only act like skin cells, you know. And when you think about it, nowadays, as many genes that we know that are dysregulated, it's like, how can we alter this expression to make them act right? You know, cancer, autoimmunity, all these big playing factors that we know, how can we control the environment to influence them to act more like they should? So, long story short, that's kind of how we got to this paper today, because this group is really telling some really interesting information. I'll try to make the talk as beginner science as possible. I know we have lots of many different expertise level careers out there. One being my Aunt Brenda. She supports me every week with this along comfortably with the talk. Okay, so again, I'm Antia. I got my PhD a few years ago in immunology, and this talks a lot to some of the things that we looked at in my prior lab with signal mechanisms. So let's get started. Okay. So this group here, Ding et al., actually is in China. They're located in China. Their patient group was about, I think, 33 females, and we'll get to why they selected females a little bit later on in the talk. But I don't know if you guys know this or not, but this institution is actually known as the Yale in China. Did you guys know that?
Mm-mm.
So this group here, or this institution, is a national research hospital. They specialize in like transplantation and medical technology, but they're known as the Yale in China. So they were initiated or founded by a Christian missionary society, as well as Yale graduate students in 1913. Am I the only one excited about that?
No, that's really cool. That's so interesting.
Yeah, it's just real. It's real interesting on how, you know, they kind of came to be, because when you think about resources and how to best tell these stories, especially with something like lupus, you know, this is kind of how it all started. Okay, without further ado, first we'll get more into the immunology of lupus, or systemic lupus erythematosus. Always a tongue twister for me.
5 years of PhD and yeah, it's a very hard name to say, I'll be honest.
So SLE, there's different types of SLEs. There's like cutaneous, there's like multiple different types. There's, I think, maybe about 5. But we'll talk more about these T cell populations. There are more than one T cell population. We'll talk about the B cell activity, as well as what epigenetics actually is in general. And how we can kind of control some of these epigenetic factors with methylation or acetylation. And then we'll go into what miRs are, or microRNA is. So first, lupus, very, 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 very nasty, nasty disease. It's an autoimmune disease that produces these autoantibodies towards self-tissue. Not really understood how that actually has came into play. They're not really sure the etiology of lupus, but this autoimmune disease breaks self-tolerance. So instead of saying, hey, you're my arm, I'm going to leave you alone, it says, hey, you're my arm, I'm going to attack you. And with lupus, it actually attacks in multiple locations. So it's not just one area, but it's systemic. So it can attack any organ of the body. So as you can see, the skin, the heart, kidneys, blood, muscles, joints, and the list goes on. It's commonly diagnosed in 15 to 44 years of age. I will say, though, that it is for a 4-to-1 ratio with pre-puberty, but when females more so grow out of puberty, it actually doubles to an 8-to-1 ratio post-puberty. So more in those 18 to 44 years of age. So again, you can see signs of lupus a little bit earlier on too. 90% of patients are women, more so African-American women. We are like 4 times greater in population than any other ethnicity. It's treated by a rheumatologist. Usually you have this present signs of autoantibodies, and they're mostly treated with hydroxychloroquine, which has been circulating all in the news today, right?
Oh, yeah.
Interesting story, though. It was serendipity to how they discovered that hydroxychloroquine actually helped to treat lupus patients. So originally we know that this drug treated malaria patients, right? And that's a parasitic disease. But it's kind of serendipity as to, oh, let's see if this works, and kind of how we're seeing today, actually, you know, right? Okay. So to
wait, just to stop there, what my idea about how hydroxychloroquine works in lupus is it destabilizes the TLRs that are in the endosomes, right?
Yes.
Yeah, because TLRs, they have a big role in signaling against. So lupus somewhat looks like a viral disease. That's a funny thing about lupus, because the cytokine response, the immune cell response, it mimics what a viral response will be like. There'll be responses against foreign DNA, and a lot of those TLRs that recognize this foreign DNA are inside the endosomes, and that's where the hydroxychloroquine helps. Yeah, that's all I wanted to add.
Yes, and that's correct. And so I had a point to make along with what you were saying, saying with that. I'll remember later on. Okay, but so SLE has a criteria. I actually had a undergraduate student work under me during my graduate studies that had lupus. Well, not had, but she has lupus as well. And man, when you really see it up close firsthand to how it actually affects patients, it really makes you that more passionate on really understanding how can we treat this, how can we understand it better to help just alleviate at least the pain in these patients. Lots of people familiarize lupus with the butterfly rash, more so on the face and rosy cheeks area. But the more and more I dive deeper into understanding just how it affects people differently, the rash is not as prevalent as I've seen it to be. I don't know if anybody else has kind of witnessed that as well.
Is that the rash that's on the face?
Right, the butterfly rash.
The butterfly rash, right.
It's not as prevalent as I originally thought. You know what I mean? Like my student, she said that she was going for some type of trip. She got out the car. I think she lightly bumped her ankle on the car something. It wasn't even, it wasn't, you know, something severe, but that led to a growing wound on her ankle that massively just increased in size week by week. And she actually, she had to go to the nurse's office to suck the fluid from it because it just wasn't healing, you know? So when you also take the drugs like hydroxychloroquine, it's an immunosuppressant. So like Jatin said, it dampens those TLR responses. So inflammation is a good thing, but because lupus causes too much inflammation, you know, hydroxychloroquine helps to lessen and dampen those inflammatory signaling. So that mixed in with just the dysregulated immune system, I guess.
Did Anthea get cut off for everybody?
Yeah, that got cut out for me as well.
Yeah, Anthea, we didn't hear your last 10 seconds.
Okay, I just said that it's just a very nasty disease to have to experience. So again, there are 11 different symptoms that can fall under the category of lupus. But if you are presented with at least 4 of these 11 things, then you're classified as having lupus. Does that make sense to everyone? Okay, so they call it the 411. Okay. So because I'm an immunologist and I like to talk all about immunology, this paper, as well as everything else, will be circled around these immunological disorders that actually happens in lupus patients. Okay.
Sorry.
Sorry? What did you say?
I think Maria is also getting disconnected.
Oh, okay.
It's fine. She'll be back.
Okay. Okay. So, because I know my sweet Aunt Brenda is listening and she knows nothing about T cells and B cells, we're going to try to teach her, as well as other beginner scientists, a little something more digestible to understand. So in lupus, right, in general, we'll just say in general, because as we all know, there's a lot more details. But in general, with lupus, you have these pathogenic CD4 positive T cells, right? And this is kind of what the paper is focusing on too. Pathogenic CD4 positive T cells like to influence these B cells. B cells produce antibodies. Antibodies are like sticky notes. They like to stick and tag on things to mount an immune response towards. So if these pathogenic CD4 positive T cells are influencing these B cells would then come along and attack multiple different organs, right? We have the skin, we have different components of the body. But in the immunology world, what we like to do is when we have dysregulation, we have a officer that comes in that likes to regulate those things. So the paper doesn't talk much about regulatory T cells, but I do want to put this in here because regulatory T cells help keep the balance between once nice T cells, CD4 positive T cells, tissue.
And I think relevant to the those T cells that you just mentioned is the CD25 and CD69 are markers of those Tregs, correct?
Yes.
No, not 69.
So
Not 69. Okay.
yes, not
CD69 is a general marker for any activated T cell.
Yeah, as well as CD25, which is also activated, but is also found on Tregs with FOXP3,
FOXP3
and others.
Yeah.
very positive. Yes.
Yeah. Okay.
So thank you. You told the grand surprise of the story after. No,
Oh, I'm so sorry.
no, I'm joking.
We have been spoiled.
I'm joking, but so again, we need to regulate these CD4 positive T cells that are now pathogenic, that are influencing these B cells to tag their little antibodies on different parts or tissues of the body to cause self-damage.
Right,
Okay.
right when we are here, I'll just plug in something. My whole proposal for my PhD is about these regulatory T cells not working as well as they should and how we can fix them. So this is very close to what I do, or
Yes.
I'm planning to do at least.
That's cool.
Okay, so next, the authors talk a lot about these epigenetic markers, and epigenetics is, just to generalize it, it's the, basically it's the, how do I describe it? It's the like secret lock and key to the genetic information, if you think about it like that. So we all know we have DNA, we all know we have chromosomes and this genetic information, but we know that it's tightly coiled and wound up to be these chromosomes. So in order to loosen these chromosomes up and to express certain genes, we have to have some epigenetic influences to actually unload or unwind these genes so that they can be expressed. So epigenetics can be affected by factors like development, the environment, the chemicals, environmental chemicals, drugs, pharmaceuticals, even aging and diet can also play a role in epigenetics and how they influence this gene activity. So they can either activate it or repress it. So when we look down further with these histone tails, the DNA wraps around the histone, kind of like a yarn and spool, or yarn and spool, and it ravels up and it suppresses that gene activity. So when the gene is ready to be expressed or used, you'll have epigenetic factors like such that tags on to these histone tails to either expose the gene or repress the gene. And here's where I want to put in methylation is a way to suppress gene activity. When a histone tail is acetylated, that causes the unraveling of these histone clusters to then express the gene. So just try to remember methylation, suppress, acetylation. What's the opposite?
Express.
Yeah.
Express.
Yeah, that's a good one. Express, repress. Okay, we should make a song about that. Okay. Lastly, for our introduction, we'll go more into these microRNAs.
Just a second, Anthea, can I intervene for a second?
Of course.
Yeah, for acetylation, if it makes it easier to remember that, acetylate, so histones are positively charged proteins, mostly because they have a lot of basic amino acids, and DNA is negatively charged. So a part of their binding is electrostatic, and acetyl groups are negatively charged. So when they are attached to histones, they saturate that positive charge, and that is one way the electrostatic interaction loosens and the DNA is more expressive, more open conformation. I actually don't know what's the point of methylation. I don't know how methylation on the root level, how is it affecting? If anybody knows, do share, please.
Yeah, that's definitely something I can share my biochemical notes on on later on. But just to keep it simplified, that's correct when you talk about the different charges. To our beginner scientists or beginner learners, again, when you talk about acetylation, we want to express the gene. Methylation is usually used to repress the gene. Okay, so we go on to microRNAs, and I won't bore you guys with all this cool information, which is kind of juxtaposing itself, right? Boring, cool information. But when you have a microRNA to then come into formation after all of the long journey it goes through with DROSA and snipping ends and collecting more information. But microRNAs are actually classically known to be non-coding. So they were actually called junk DNA, so it was something that no one took serious. It's just like, oh, it's a bunch of snippets, so let me go ahead and like bypass that. But it actually has a huge role in how we express gene activity or even repress gene activity. So these small non-encoding RNAs, they're about maybe 22 nucleotides in length. They're involved in gene regulation. So to simplify it, if you have a microRNA and it sits on top of your promoter region, it can code to degrade the gene of interest. If you have the microRNA or the miR that sits more um, past the promoter region, it can block that translation. So it can either block it and cause for a dysregulated protein expression later on, or it just blocks it and suppresses it totally. So in terms of like autoimmunity, lots of people in the epigenetic world look at microRNAs to see if they are actually suppressing what we need to be positive expression in certain situations for different disease cascades. So a bit of background for this paper. These authors wanted to they first looked at healthy cells, or previous literature also indicated that in healthy CD4 positive T cells, when you take out miR-142, 3P, 5P, and they come into a very long journey of how they actually got here, but I'll fast forward it to the point that if you take out this miR-142 group, it will significantly increase IL-10, IL-10, CD84, and also the SAP family proteins. In general, because the paper doesn't touch a whole lot on it later, but these group of proteins are pro-inflammatory and they're heavily implicated in lupus pathology. So when you see IL-10, CD84, and SAP proteins, as well as other proteins together, these are heavy indicators of inflammatory signalings in lupus patients. In addition to that, you have increased T cell activity and increased B cell activity. And remember before, in my little illustration, we know that these T cells can influence these B cells to then go and express these antibodies to tag multiple organ tissues that causes self-damage in lupus patients. Take away miR, it then won't have the opportunity to suppress these pro-inflammatory signalings. Opposite to which the authors looked at lupus CD4 positive T cells or SLE positive T cells, and they noticed that it's actually decreased. So miR-142 group is actually decreased in SLE patients. And again, this occurs in lupus patients, CD4 positive T cells. So there's no outside stimulation, there's no interferences with it. They're noticing that there is less miR-142 in lupus patients. So then it becomes the question, how can we, what type of effects will affect this if we actually increase miR expression? So again, remember microRNAs sit on top of gene segments or promoter regions, and they can either degrade or suppress expression, right? And we kind of want that in lupus patients because we have hyperactivity of these IL-10, 84 SAP proteins, T cells and B cells. So we're saying, hey, or sorry, the authors, I'm including myself now, but the authors are saying, what if we overexpress miR? Can we then dampen these inflammatory and excessive lymphocyte responses? So what they did was they experimentally performed that if you jack up miR-142, these expressions should go down. Now, brings us back to the original question, because Jatin likes a good meme. The question is how? How is miR naturally occurring in lupus patients, CD4 positive T cells? How is this actually being decreased? How is this regulator not being regulated? Or how is the regulator not regulating other immune responses like healthy cells? So that brings in our star pupil, Bcl6. And Bcl6 is a B cell lymphoma 6 protein. It has a conserved zinc finger transcript, and it's a transcription factor. So that means that this Bcl6 is a protein that initiates gene expression. That's what a transcription factor is. It's expressed in mostly all cells, mostly in B cells and T cells in the germinal center, but it's also critical for follicular T cell or follicular helper T cell cell formation, right, Jiten?
Yeah, Anthea, can you just give us a primer on what are these follicular helper T cells?
A primer?
Like, what are they? Or not the PCR primers. I mean, just what are they? What are these follicular helper T cells?
The same thing to give a shameless plug on our last podcast we did a few weeks ago.
If you want to know in more bigger detail, do check our last Buddysode where we discussed the follicular helper cells. But for now, just for a brief introduction, these are helper T cells. These are a subset of helper T cells that go inside secondary lymphoid organs, as in they are already probably in the lymphoid organs. They go and help B cells produce antibodies. And they are very relevant here since Anthea said that T cells helping B cells is one of the reasons the disease spreads.
Yes. So it's logical enough to come to Bcl6 to actually have an influence because, like Jatin said, these T follicular helper cells influence B cell expression of antibodies. They do this actually simultaneously while repressing the rest of the T helper subset populations. Okay, so it also interacts with different co-repressors. It's known as a histone modifier. And this study actually reveals that this Bcl6 can bind to this miR-142 promoter region, and we'll look more further into how and why and things like that too. But the author suspected that this decrease in miR-142 group expression in SLE patients was abnormal, was due to the abnormal expression of Bcl6. So we get into the meat of the paper, figure 1. Okay, so the first question in most researchers' mind is we have to actually validate or actually have to set the stage that our star player, Bcl6, is actually having some differential expressions in lupus patients versus just, I hate saying the word healthy because we all have stuff going on, but non-lupus patients, but we'll use the word healthy for now. And so in this figure, we see that if you take the gene expression or if you probe for the gene expression of Bcl6, that is significantly higher in lupus patients. They then further go on to show that both in gene and protein. How convinced are you, Jatin, with these Western blots?
I'm all in. Although GAPDH, so there was a paper about GAPDH. I think we have mentioned this or not, but GAPDH is not a very good control because it's a glycolytic enzyme, and especially in immune cells, glycolysis is upregulated and downregulated depending on inflammation. So it's just that you could have a higher GAP-DH if there is, in effector T cells, there's higher GAP-DH because they rely more on glycolysis. It's just that GAP-DH is not a very good marker. But
So
I'm okay though with this.
So what would be your ideal housekeeper gene then to do this comparison?
It's
for
actually a
this?
pretty difficult task to choose a housekeeping
Right?
gene. Yeah,
Yeah.
it is. I have used this this amino acid transferase enzyme, or it's a mutase enzyme called PPIA, prolyl something. It's something to do with amino acid metabolism, and that seems to be a good enzyme for control. But yeah, it changes with some enzyme, because definitely some gene A is affecting its transcription, right? And we don't know how all that is connected in this web.
Yeah, and this paper. I'm sorry, I'm done.
No, go ahead. Go ahead.
Well, I was going to say this. The GAP-DH is pretty significant because when they do the delta PCRs, they're using GAP-DH as that first normalization.
Well, here at my university, we mostly used as a control actin protein. So I don't know. I don't know if it's good for T cells, but sometimes we use this.
Yeah, we also generally used actin and tubulin for Western blots.
Yeah, I'll be honest, I also use actin, but it's just that it's good to know that actin is also not a good control. Also, especially again in cells that are constantly dividing, because actin, the amount of actin changes in the cell during cell division, and I'm probably sure it changes for other reasons as well. But then that's our limitation. We can't help it.
But when you think about how to analyze a data set like this, everything is always relative to each other, right?
Yeah.
So even if there were higher expressions of GAP-DH or actin or whatever housekeeping gene you have, it still should be relative towards the expression that you're looking for amongst the group in total. So I agree that people definitely should do more sound digging on the best housekeeping gene. The skin has a totally different like rules to which housekeeping gene would be the best. But I think the most important part to think about is that as long as it's relative towards the general expression of it, and you're doing your calculations pretty tight, that we should at least get useful information out of it, which I think this data shows too.
For sure.
Yeah. So next, they analyzed this correlation, which I actually really like these figures here for C and D, because they did a correlation analysis, which I think I should have did more of when I was writing papers.
And yeah, I want to thank you for this paper, because we just submitted our paper this week, and
yes, I got the email.
And it had 5 figures. So Anthea is a co-author on that. We had 5 figures before. Then I saw this paper, and then I realized these guys are doing correlations with, in this case, Bcl6 and anti-double-stranded DNA antibodies. So I did a correlation of ANA, anti-DNA antibodies, and FOXP3 expression in some of the cells we are doing, and we found significant correlations that added to a 6th figure just thanks to this paper. So now it's 6 figures in my paper. Thanks, Cynthia.
Maybe you can bump my name up then to.
I'll add that in acknowledgements. Suggested a paper.
Acknowledgements. There you go.
Okay, so the authors wanted to analyze the correlation between Bcl6, I keep wanting to say BL6, but Bcl6 expression and clinical pathology parameters. So they looked at both the double-stranded DNA as well as the SLE scoring index. So just a brief background about that. The higher the physical severities, as well as like antibody production, double-stranded antibody production as well too. But the higher the severity of it, the higher the index goes. So when you create these DAI scores, this actually is direct correlation of the severity or the severity index to the disease activity. We also created something similar to this in our psoriasis papers, where we had our mice to develop. So we created our own severity index. It's called PASI in psoriasis patients, but we created our own PASI index for the mice that correlates directly with the actual human severity. So very useful thing for physicians to kind of gauge and scale the severity of their patients as well too. But they did this correlation analysis, and this data shows that the expression levels of Bcl6. Oh my, sorry about that. Did you guys hear that crash?
Yep.
Oh man, sorry. Okay. So the expression levels of Bcl6 was actually positively correlated with the double-stranded DNA in the SLE score. So if you look down further, it says the relative Bcl6 protein level. So the higher the Bcl6 protein level, the higher the titration of the double-stranded DNA is, as well as the SLDAI score. They then went on to want to validate that as they stimulate the TCR or these T cells, that there will be significant increases with Bcl6 expression in normal cells to start off with. So, you know, before we get into just the whole scope of lupus patients, they want to set the stage up to show you that in normal cells, this is how BCL is working and playing a role. And as the time goes up, you see that it increases with TCR stimulation. Here, when I saw this data, because I've done a lot of probing for transcription factors, I usually see transcription factors earlier on. Like literally, you can look at the cells and you'll get some type of transcription factor activity. Like it seems like that. So when I was probing for some of the STAT proteins, at 30 minutes, boom, there's STAT expression. You know, as soon as we stimulate these cells, there's STAT expression. So it's pretty interesting to see that this Bcl6 is actually more at 6, 12, and 24 hours. I'd like to see what it looks like, and then at 0 time point. So just wondering how it looks at a little bit earlier on. Okay.
But STAT proteins are, they're known for transiently being expressed. So they go up very quickly. And then they, by the way, in case anybody doesn't realize, STATs, we are talking about the Janus kinase and STAT signaling. Some cytokines take that pathway. And yeah, we both worked on that. She is in the same lab as I'm working right now. She graduated from there. That's how we know each other.
Okay. So lastly, the authors wanted to determine the relationship between Bcl6 and the actual activation of CD4 positive cells. So to move from a point of just validating correlation, the authors are trying to drive the point home that there is an actual causation, right? So the difference between correlation and causation, it's not just a coincidence, but this is an actual player in this activation of some of these TCR active or T cell activated markers like CD25 and CD29. I don't know if they were expecting these results. It's also activated functionally as well. So to look at the healthy cells, they wanted to give insight on SLE. Did my connection go out? No.
It did for a second.
Okay.
Yeah.
Okay. So to look at healthy cells, they gave insight in SLE, and it shows the downregulation of Bcl6 expression upon this TCR or T cell activation.
And yeah, you know, in F, if you go back, just in F, this PRS, was this a plasmid expressing Bcl6? Or is
so
this
the PRS was the empty vector control,
right.
And then the PRS Bcl6 is the one that upregulates
Bcl6. Yeah.
It's very strange that adding more BCL6 reduces CD69 expression. That's opposite of what we discussed in our podcast there, where increased CD69 expression went hand in hand with more BCL6.
Right. So
Maybe something that maybe it's a feedback.
Back in the discussion, they kind of were surprised with these results. Okay. So next, the authors wanted to look at the actual effects of Bcl6 on miR-142 group in SLE patients. So they want to cut to the chase. They don't want to keep, padding around with, oh, does Bcl6 have any effects on miR? So they want to go right into it. They did another correlation plot where it looks at the gene expression of the miR-142 group and how it correlates with Bcl6 protein levels. They did the same thing for each subgroup. And they show that as the BCL levels increase, you have this decrease with the miR-142 in both subgroups. So as the Bcl6 increases, these miRs decrease. So they're playing into the narrative that there's a lack of regulation in the overregulated T cells stated before, because of the suppression of 142, which we already know from the beginning literature. Next,
Anthea, 1 second. Yeah. So I was too lazy to find out what's the difference between 3P and 5P. Do you know? What is it? Is it like alternate spliced forms?
Yeah, so I didn't want to go into grave detail of what each one was and did, but they're both a part of the miR-142 group. They're just different components of it.
Okay.
Yeah, here, I'll send you the paper on it. Yeah, I like to keep them together, but they do separate it out, but they're just different fragments of the miR-142.
Okay.
Okay. So here in C, the authors wanted to investigate the effects of Bcl6 on the miR expression. So again, they put it into that plasmic vector. They see that it actually is increasing this Bcl6 expression, so the transfection worked. However, when you transfect these cells into, I think these are just normal. Yeah, these are normal. These are normal CD4 positive T cells, and these are SLE patient CD4 positive T cells. And when you are transfecting the Bcl6, this miR expression goes down, which is kind of what we see here anyway, but it's actually in reverse to SLE patients. So it's actually decreasing these miR-142 subgroups. So this is an actual more direct correlation. I'll get more into it actually directly playing a role in these miRs, but this is a more definitive correlation that Bcl6 is actually influencing these miR-142 groups. Okay.
Excuse me, isn't this PRS plasmid actually a disruption plasmid? I mean, doesn't this plasmid interfere with Bcl6 expression?
Yes.
Wait, is it?
Yeah. I'm sorry.
Okay. Hold on.
I'm sorry.
Because in that case, it makes sense.
Right. I didn't explain that thoroughly. Sorry. So when they're putting it into Bcl6 into this PRS, it's to decrease that Bcl6 expression. So when Bcl6 is decreased, it's showing the reverse effects that it's increased in miR-142.
Okay.
Okay, so in that case, the reduction in CD69 and 25, that is normal because it's disrupting Bcl6. Okay.
Right.
Yeah, I have a very bad habit when I make notes. I think something is obvious, so I don't write it down.
Right, and then the next day you look over it, you're like, oh wait.
Yeah,
I'll just read directly off the chart, and I'll be able to figure it out. I'm so smart.
Yeah.
Yeah.
Thanks for the catch.
Yes.
Okay, so this is actually one of my favorite figures because I don't, if anybody's ever submitted a paper before and got rejected and they say this is correlation, how do you actually know that this is working? What is the point of this? Like all the reviews that you get, it kind of is head scratching. And I think this is why this paper has gotten as much publicity as it has because it directly shows that they're not playing around anymore. They don't want to show correlation. They want direct context. So it's showing that Bcl6 is having a direct effect on miR-142 in these SLE patients, CD4 positive T cells. So if you add in a probe for Bcl6, you see that it's present upstream of the translational region. So you see the presence of, I'm sorry, now I'm getting frazzled. Okay, yeah, so
these are always confusing. These precipitation experiments, you don't know what are they pulling it down with and then what are they probing it with. I always get confused with these.
Yeah.
So
okay. Go ahead.
Oh no, I think they're pulling down Bcl6 and then they are going for PCR with the to promote proxy. Yeah, and they're going for the PCR for miR-142.
I was going to ask too if anybody wants to give more details on chip analysis.
I can if nobody else can.
Yeah, go for it. I don't do any chip, so.
Okay, I have never done chip, but the simple the way it's done is you probe for you have some sort of solid support like beads that have an antibody against, in this case, a transcription factor Bcl6. So they will pull down all the Bcl6 from a cellular extract. That pull down material should only have Bcl6 and all those things that were attached or were interacting with Bcl6. Next, on that pulled down extract, you perform something like, in this case, a PCR. So only if Bcl6 was interacting with DNA around that miR-142 region, it would have got pulled down with it, and now it would show in the PCR.
Yes. One other thing too, I don't know if anybody has done an enrichment assay. But I was trying to figure out more so of how they get this relative Bcl6 enrichment, they call it, and they go on to talk more about this enrichment later on. I'm not sure if that means that they're culturing cells with Bcl6. I don't think it would just be called that, but I don't know. I'm struggling.
I think it means that. Let me see.
Is that just that they were enriching the CD4 cells, or that they were enriching Bcl6?
So they were enriching Bcl6 in CD4 T cells, but when enrichment is used, I'm thinking like, is this just, are you saying stimulating these cells, or is it?
I think they just mean when you pull down, so you have a fraction of, you have all of the lysate of the cells which have everything, and then when you pull down just Bcl6, so now you have this enriched fraction which has Bcl6, and they found that more of Bcl6 from healthy patients bound to this miR-142 promoter region compared to from healthy. Yeah, more in SLE patients than healthy.
So they're just using enrichment as a synonym for like isolated or pulled down from, as opposed to
like that.
Enriching like a splenocyte culture or something like that. I think.
B is actually chip PCR, so not just amount of Bcl6, but the amount of Bcl6 that is attached, or probably the amount of Bcl6 that are attached to the miR-142. I could be wrong though.
Okay, cool. Thank
you.
This
is another one of that experiment where you see and you think, I understand it. Then when you when you sit
You
to
have to
discuss
talk it out. Yeah.
Yeah.
Right. I'm glad I have the support of you guys. Okay. All right. So moving along, the authors are now asking the question, how does Bcl6 actually regulate this miR-142 expression, since we know that it binds? What is it altering? We know that it's having a direct effect here because they are bound together, but how is this altering this gene expression as time goes by? As we see it, it's being decreased in lupus CD4 positive T cells. So they show here in A and B that this BCL regulates by altering histone modification. So it's actually H3K27 methylated 3. And when you see things like ME3 or AC, so forth, this is just saying that this histone, or the third histone, is methylated in this region, the K27 region. So this, or in the third spot, or does
No,
that
I think
mean
it's a trimethylation. It's a trimethylation. So this spot,
Yeah,
S3K27, gets three methylates.
That's right. So the three means that they're methylated 3 times.
Yeah.
Okay, or there's three methyl groups. Let me not say 3 times.
So hold on, they had this PRS, which is a disrupting plasmid, and what's the PCMV6?
That's
the one that they plug into, I think, the wild type CD4 cells to induce Bcl6. Right.
Is it,
they use so many plasmid vectors.
Plasmids and
It's like
yeah, it was written that that normal CD4 plus cells were transfected with this PCM.
okay, so I think they're like trying to replicate.
They're
what if?
mimicking.
Yeah.
Exactly. Yeah. So they're inducing SLE like conditions into the normal healthy CD4 T cells.
Is this cytomegalovirus? Tanu, do you know
Yes.
why?
Like that, yes.
Like what's?
I think so.
I know sometimes they use lenti retro. Is there any benefit of using CMV? Or it's just can use anything?
I haven't heard of using CMVs, like I haven't read any article about it, but CMV stands for it. But PCMV, I don't know how they're making a plasmid out of it. No.
Or it could be some,
experience.
or is it completely different and we're just mixing it up?
Yeah, maybe.
I think that also CMV sometimes in some articles stands for cauliflower mosaic virus, but it's plant virus. So I don't know. It probably shouldn't be in animal or human
Wait
studies.
a second. In the materials, it says plasmid CMV. So I think it has nothing to do with viruses in that case.
I think it just has the promoter from CMV.
Okay. And is that the cytomegalovirus or the cauliflower CMV?
Does not explain that. Cytomegalovirus. It's an ad gene. It says mammalian.
Isn't this promoter from cytomegalovirus like a very strong promoter? So like
it should be.
It induces
That's what I was thinking, right?
Yeah.
Mm-hmm.
Something
And
like
especially
that, I
when
think.
you're dealing with these mammalian cells.
Mm-hmm.
I think it's appropriate.
This is the right time for me to take out that card that says I'm not a virologist, so I have no idea.
You see the two immunology people are just quiet.
I keep a lot of those cards. Very handy.
Well, anytime I see plasmid or vectors and things like that, I just think, oh, okay, well, we're about to just inject something that we want to be there.
Yeah, we don't need to hear that. Not
Right.
right.
And the method that they used wasn't even injection; it was electroporation. So they shocked all the cells to get those in. Yeah.
Oh yeah, because you can excite the membrane a bit, right, to cause semi-permeation.
Exactly. Yeah. So you shock the cells. The Amaxa nucleoeffector is actually a pretty cool instrument to use. So you have these little cuvettes, and you just take your cells, you pop them in the cuvettes, you pop in your plasmid, and then you just shock it. You literally, like, they're programmed already for the conditions, and then you just press the button, and it turns, and then it goes, bzz, 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 and then it turns back, and then you take out your cells, and then you throw them in your media.
Oh wow!
And that's it.
How do you select for the cells that didn't accept
Get transfected?
whatever? Yeah.
Yeah. So if you're looking at like a transient transfection, you just have to rely on the fact that you're going to have increased gene expression. But if you're doing like a stable culture, it looks like this CMV plasmid has both kanamycin and neomycin resistance genes. So you can always do a selection if you wanted to grow up like a stable culture, but odds are good if they're using electroporation that they're just looking at a transient transfection, and especially for like anything with microRNAs, it's going to be transient. So you just have to rely on that initial whatever you pump into it is going to have enough of an effect to be able to see something downstream.
Okay, cool. That's cool to know. That's I know we're extending our time a bit, so I'll try to hurry up. But just something I've picked up on after graduating and going into the real world of life. That's a really useful technique to have, as well as knowing the immunology and things like that too. Because when you go into actual therapeutic targets in the industry and maybe some of these pharmaceutical companies, that's exactly the type of skill set that they're looking for when testing their drugs. So I think that's something to keep in mind too.
Yeah. Although I also think that these are things that can be learned very easily, as long it shouldn't be very difficult, right, to be running these experiments. But
I think that the most difficult part is this selection markers.
Mm-hmm.
We did something like this with yeast during one of our classes, and the most difficult part was to somehow design this plasmid that it has those parts that, I don't know, induce resistance to some antibiotics and things like that. So it was the most difficult part, actually.
that screening part, yeah, I've never had to screen. I'm very happy I've never had to screen. It looks like a lot of work.
Yeah.
Okay. So I'm just going to cap up this figure, if you guys don't mind. In total, they just wanted to know how, since this Bcl6 is binding exactly to this miR portion, what is it regulating? And they indicated with this figure several histone modification areas that are influencing this miR region in SLE patients, even compared to control regions. So again, if you're seeing acetylation or AC at the end, that's usually for expression of the gene, and methylation is more for repression of that gene. So together, these results suggest that Bcl6 is actually suppressing the expression of miR-142 group by increasing this H3K27 methylated 3 levels and reducing those expressing modifier segments of H3K9 and 14 AC in CD4 positive T cells.
It's such a neat experiment.
Yeah, it is. And it's so like straight to the point too. You know how I love flow cytometry, but sometimes it's so subjective.
Yeah, you make your own data.
Yeah. It can be so subjective that it's like, oh my gosh. So when you just stick to, is it there? Is it not? Is binding? Yes. So now what does that mean? We can get to the point of the mechanisms. So
yeah, I just want to, if you go back, I just want to highlight something to the viewers in case anybody missed it. So in the section A, they increase it in healthy, and they show that it looks exactly the difference between the healthy and the SLE patients. And then they disrupt it, and they find that this H3K27ME3 is reduced. So they are drawing a very clear line that this is affecting. Could be direct, could be indirect, but this Bcl6 is affecting by itself.
Mm-hmm.
So it's a very nice experiment. Anyway,
Yeah.
I'm just amazed.
Great model to use, and they were able to show this like double whammy where they have an increase in the methylation and a decrease in acetylation, and then it's just like
and I'm not an epigeneticist, but I feel like this is so this is so just like, what's the word, so straight to the point. You know, it's there or it's not. It's going to open, it's going to close, what's binding there. And this is a great way to kind of just really look at any disease model when you think about it. Not just lupus, but a lot of autoimmune diseases have these miR components to it, but people aren't too sure on how it's being regulated or if it's actually even doing anything towards specific cells. This is also talking about specific cell populations too.
Yeah.
You know, it's not just a whole pooled cells. This is saying no, T cells, right? CD4 positive T cells.
CD4, yeah, even more specific. Yeah.
Reviewer 2 will look at this figure and he will be very sad.
Happy. No,
he'll be very sad.
He's got nothing to say.
Because
nothing
you cannot
to say.
complain. Come on. Yeah. Got it.
Yeah. No, he could say that the thickness of the bars are not consistent throughout.
Oh, yeah.
The error bar is very high too.
Rerun these experiments.
My reviewer for my bachelor's thesis complained about the color of the bars. So, you know, he was just horrible.
I mean, it does look like this was made in Excel and not in GraphPad.
That's true.
For shame. And sometimes in the past, like I've removed all bar significance. You know how you have like the bar and it's like the stars and stuff. Some people just put p-values and say it over there. So I've seen lengthy complaints. I hate the p-values. Use the asterisks.
Right?
God, especially when they have the asterisks at the end of the figure and they're like, what, like p is less than 0.0001 is 4 asterisks, but then they go ahead and put a p-value anyway. It's like, I don't want to look at a p-value. I like little stars. It's way easier.
That's so funny. Okay, I'm going to just try to cap up these figures. I'm sorry if anybody is going to, wants to point out something specific that they found interesting, feel free to jump in. But I'd like to just cap up the remaining figures just for the sake of time. But to explain that the authors, going back a bit, they see that it binds to the miR region. Bcl6 binds to the miR region, and it's playing some role in histone modifications with methylation and acetylation, but they want to explain how. So they picked up these influencers through string database. Anybody's ever used string database? I love string.
I find it slightly confusing, but yeah, I can imagine using it someday if I do this kind of work.
Yeah. So, oh my gosh, Dr. DeCracy would be disappointed that you don't remember string just him.
And I'm very glad she's not watching this. If she was our bioinformatics professor, she's really good at what she does. But unfortunately, I was not very good at listening.
Oh man, yeah. So I played around with bioinformatics too, but string database is where you can identify proteins that might interact with the protein of interest. So in this case, it's Bcl6. What it does is it shows from published literature. So it's not just something, just thrown out there and said, hey, go look at this protein, this protein, this protein. It's actually a database that computes proteins that are published that have some correlation. And it actually tells you the strength of that correlation too. So it'll say, this is closely related or this is less closely related, but it still has some type of pipeline through it. But string database, when they use this, indicated that TP53 and FOXO3 and HDAC5 is related with this Bcl6 player, right? So then they noticed that this HDAC5 is more closely related to histone deacetylation. So that's kind of how they use HDAC5 as well as the EZH2 to move forward with how these components are helping to assist the methylation and acetylation in these histone modifiers that we saw previously. Like, that makes sense to everybody. Anybody want to elaborate a bit more on this?
On
Oh, well
histone
said.
modifying enzymes?
Well, just this figure in general is looking at the players that are heavily associated with Bcl6, and these were picked up again by this string database.
I just realized I did not write what's EZH2. The HDAC is the histone deacetylase. What kind of enzyme is this EZH2?
So the EZH2 actually in the literature has some relations with the follicular lymphoma, but it's a histone lysine methyltransferase enzyme.
Okay.
Well, there we go. That makes a whole lot of sense.
That
if we
makes
look
sense.
back
It's a
at,
methylase.
yeah, exactly, at our
Methyltransferase.
lysine methylation.
Yeah.
And it was named because it has some role in the development of the organism. So that's how it was named initially,
Right.
but it's not named after its enzymatic activity.
Right. So they're touching on the point of methylation of these histone modifications and the acetylation of it. So they located this HDAC and HDAC5 and the EZH2 is playing a role in the deacetylation and methylation of it.
And both of these enzymes will result in a modification that leaves the DNA less open or in a closed state.
Yeah. Okay.
All right.
I'm going to skip forward because we are 10 minutes in. And lastly, the big question overall is, all this stuff is fine and dandy, but does it work in patients? Right? Like, I think that's the end goal to everyone's experiment. I don't care how far from the disease model you are, it always draws back to, can this help people? So the question is, can this mechanism to target, can this mechanism of this Bcl6 and this miR-142 region be a target to actually work in SLE patients? So what they did was they looked at these T cell markers like CD40 ligand, icosapent IL-21, and the author discovered that when you knock down Bcl6 expression, it alleviates the self-reactivity of SLE CD4 positive T cells. So to do this, they wanted to determine the effects of the downregulation of Bcl6 expression in SLE CD4 T cell function. So when you look at function, you usually look at these transmembrane markers or activation or cytokine expression, which you see here in A, and it shows that both in the T cells, that the mRNA and protein expression levels of CD40 ligand, ICOS, and IL-21 in lupus patients were also detected. So that's a big tongue twister, but what they expected was that they observed a significant decrease in CD40 ICOS.
L for CD-L, yeah.
Yes, CD40L, because CD40 is on APCs, but the CD40 ligand is on T cells. So these are direct markers of CD4 positive T cells, and they are significantly decreased. When they go on to look at, let's see, I'm sorry, I have a bunch of notes here. So furthermore, they wanted to look at the CD4 positive T cells in which Bcl6 expression was knocked down. They co-cultured with purified B cells. the IG levels in the culture supernatants were measured, and then they performed an ELISA to look at the output expression of it. So there's IgG antibody production, which again are produced by the B cells. And we know that there's a lot of IgG being produced in SLE patients too. With this antibody production, these Bcl6 interference groups was lower than that in the negative controls.
Yeah, I want to just make one point here that these markers that they have chosen, they're very specific markers, not just for any CD4 positive T cell. They are markers that define follicular helper T cells, because CD40L is important for helping B cells. ICOS is required for getting help from B cells, and IL-21 is required for both B cells and the T follicular helper cells. Do you guys remember that
Yes.
that phenomenon called somatic hypermutation? Have you ever heard about this thing? So somatic hypermutation, which allows the only cell type in our body which can go through that kind of mutation and still not die, B cells, they use an enzyme called activation-induced cytidine deaminase, AICD. And that enzyme is stimulated by IL-21 that T follicular helper cells make. So without IL-21, B cells would never be able to go, apart from a lot of things, they would not be able to go through hypermutation. That's why this is very important. And they're effectively so, and Bcl6 itself is a marker for T follicular helper cells, so it makes sense. So they're effectively showing that this help towards B cells is reduced when you remove Bcl6 from the equation, or reduce it. They say knock down, which is the right word to use because they're not completely removing it.
Completely.
Right.
Yeah. Thanks so much, Uten, for that. That really ties the point home earlier when we talked about how Bcl6 is more indicative towards T follicular helper T cells, and how this influence is playing a role with the suppression of we now see in lupus CD4 positive T cells. So I just really like how they capped everything.
Yeah. Initially, my hypothesis, so I work on something like a drug, and we found that our B cells were reduced. So my initial hypothesis was that it was acting on IL-21. The problem was I never got to test that hypothesis because we quickly moved to regulatory T cells. But yeah, that's why I had to read a lot about IL-21. That's the only reason.
Cool. So just to recap, miR-142 group was significantly downregulated in lupus CD4 positive T cells compared with healthy cell expression of Bcl6. We know that it directly binds to the miR-142 promoter region. It inhibits miR-142 expression by increasing these histone modifiers in SLE CD4 positive T cells. And when you activate TCR or T cells, this could significantly upregulate Bcl6 in normal CD4 positive T cells, which is something I don't think they kind of knew to begin with a bit. It seemed like the context of the literature seemed a bit like they were a bit surprised. But the levels of Bcl6 is having an impact on these histone modifiers by EZH2 and HDAC5, so we know the exact place. So this is just so great because they don't leave us on a cliffhanger, right? They're not saying like, something is playing a role in modifying these histones. They're actually going into it and saying, we know what it is. Let's go ahead and target it. Then the data suggests that the increased expression of Bcl6 is the main cause of reduced expression of miR-142 in CD4 positive T cells in SLE patients. So I will close with that. I do want to add in a few remarks that I had while reading the paper. So lupus is a very dear, not friend of mine, but it's a very dear disease that I just find significantly just challenging but interesting because it's an autoimmune disease, but it's an autoimmune disease for the masses. And I mean that by saying, when you think of other autoimmune diseases, lupus is a model that either has a mirrored effect in other autoimmune diseases, or it has the opposite mirrored effect of other autoimmune diseases, but it's still a central model to ask a plethora of questions in other autoimmune diseases. Case in point, psoriasis. So lupus patients are light sensitive. You don't want to shine bright lights or go outside for extended period of times if you have lupus. However, in psoriasis, light therapy is a thing. Like, they literally have devices, like little compact mirror device, it looks like a mirror, or it's as small as a mirror that you can fit in your purse, and you can shine the light over the affected area, and it'll cause DNA damage so that it promotes the system to heal it. That's in psoriasis. It's not the same thing in lupus. It's actually the direct opposite effect of. So if you want to understand how the skin works in psoriasis, you can look at lupus for its opposite effect. Or if you want to look at any other excessive antibody production type autoimmune diseases, bam, you have it again in this model. But one of the comments I did have to make was that I know that this came from a national research hospital in China, but if you look at the demographics of lupus, it's Black women,
Yep.
And
I was going to ask that, and I wasn't sure how to ask that. So
no, I definitely was going to ask that. But when you think about the demographics of lupus, it's 4 times greater in Black women than it is any other population, right? And then when you think about epigenetic factors, we know that the environment, age, food, medicine, anything can offset epigenetic effects. So all of this is super great information. I love the way that they set up the experiments. However, how does it address actual representative patient population groups of lupus, which are not what we see in this data set?
I just kind of to add to that, we note that over half of the patients here are taking some sort of medication for their lupus. How is this modulating their immune response? Is there some downstream, is prednisone doing something to the Bcl6 at some level, or messing with the histones, or something downstream?
So I've also participated on a court group. It's a clinical translation group during my postdoc, where I worked with physician scientists, and we were the basic scientists, and we had healthcare workers and so forth. And we would all do this big meeting about just maybe indicating biomarkers or for patients to turn into mouse models, or we would look at biomarkers that we can target for therapeutic uses. So it's this big old, meeting that we used to have. But one question I asked, because I'm not a physician, is that in something like psoriasis, there's a huge depression factor, right? Like they take antidepressants because it's a huge mental challenge that goes undescribed pretty much. So I said, these antidepressants can also have an effect on, the skin inflammation. So as a physician, would you take the patient off of an antidepressant to treat the disease? So when you ask the question about, like, these patients were medicated and they're taking their these drugs and things like that too, I think in some point, and the answer is no, they don't take them off of antidepressants.
Yeah, of course, right?
Yeah. Yeah.
I think it's similar with some of the initial steps of clinical trials, where they don't use exactly a placebo, but they just leave them on their current medication, and they just use on some patients, they just use those new proposed medications. So I think it will be similar.
For autoimmune diseases and cancers, it's very hard to have a placebo, because are you going to let them die? You're going to have to put them in something.
But one thing with something like lupus is that they do have a remission period. So I'm not sure how frequent they may take medication. I'm sure it's daily. I know for a fact actually it's daily. But they go through a remission period where I'm just wondering, is that a window where you can get more baseline, tests in or something like that to actually see what the native state of the disease is besides medication. But hey, this is more information than none, right? But I appreciate you guys helping me out with this paper. It's been really fun.
Great
And
job.
I do want to add
great
just
paper.
last point, since we talked a lot about epigenetics and we found these two enzymes, EZH2 and the HDAC. So up to my whatever knowledge I have about epigenetics, these are not enzymes specific to locations. Like this enzyme could be working in 100 different places at the same time. So it's possible that a lot of effects that Bcl6 has could be because of this enzyme, and it's affecting epigenetics all over the place. And this one miR-142 is just a part of it. So I find this as a challenge when studying epigenetics, because how do you know where all is it affecting?
It's all related.
Yeah, it's such a huge web.
MicroRNAs themselves are so promiscuous.
Yeah, they are also not that specific. They actually do bind with that, what is it called, some shabby binding where they just somewhat similar, somewhat complementary RNA, and that's enough for them to grab onto it.
Yeah, you depending on the miR that you're working with, you don't need complete complementarity, and when you're working with something that's 23 nucleotides long, you're going to get promiscuity. If you're working in some kind of modeling, you can use an siRNA. You can validate the function of a miR, but yeah, definitely interesting when you're looking at these kind of promiscuous factors.
Yeah.
I wanted to do a bisulfate methylation PCR.
Bisulfite, PCR.
I wanted to try to get one of those in before graduating, but I left all my samples to Jatin, so maybe he can.
I am actually proposing to do bisulfite sequencing. For anybody who doesn't know, or PCR, it's a way to check for methylation.
Yep.
in DNA.
Yeah, it's actually pretty straightforward. But
yeah, it's pretty straightforward. But yeah, I'll be doing that. Don't worry, Anthea. I'll do it for you.
Thank you. I appreciate that.
Oh, before we end the meeting, you guys, anybody wants to volunteer for presenting in the next meeting? If you're not sure yet, you can tell me later in the group meet.
When are we looking to do it?
2 weeks? From now on, I would prefer to have it every other week. So twice a month. I think that makes it periodic. We all know when it's happening, and it's also just frequent enough to not forget about it, and not frequent enough to interfere with our work.
Yeah, I'll gladly take it. Present that. Likely be in the context of viral immunology. So prepare
perfect.
yourself.
Go easy on us.
No, don't, because I'm expecting to learn a lot. Lately, I've been interested into viruses.
Yeah, well, I mean, you can't escape from hearing about viruses everywhere you go now. So yeah. I'm actually sick of hearing so much about respiratory viruses. So I don't even get a break. So I come home from lab where I work on respiratory viruses, and all I see is respiratory viruses. So
exactly.
All right, then it's decided. Harris is presenting on which date is it exactly? It's 5th of September, and we'll meet at the same time, 10 a.m.
Okay.
That's not Labor Day weekend, is it?
Yes.
Oh, I don't know. Is that? Oh,
yes.
Wait. So is that? I am still. I've been in this country for 3 years, and I still don't know why some holidays are. Yeah, so I forget these.
Labor Day,
I don't know, man.
We can do it.
Okay, so that's Labor Day weekend. So probably a good idea not to do that weekend because it's holiday. Okay. So if we could do the 12th,
oh yeah,
that would be better.
Okay, 12th August, September then. It is.
Are we still on YouTube?
Yeah, we are still on YouTube. I'm going to end the YouTube cast right now. Thank you all for watching, guys. So I will turn it off for YouTube. Not on YouTube.